How life started on Earth. A journey through deep time to the dawn of biology. First, the greatest mystery. Look around you. Everywhere you turn, life surrounds you in spectacular abundance. The trees reaching toward the sky, their leaves rustling in the breeze. The birds singing from hidden branches. The insects crawling through the grass. Bacteria living on your skin, in your gut, in the soil beneath your feet. Life is everywhere, in forms so diverse and numerous that we have yet to catalog them all, despite centuries of trying. Yet there was a time, not so long ago in the grand scheme of things, when this planet was utterly devoid of life. A time when the Earth was a ball of molten rock, bombarded by asteroids, wreathed in toxic gases, and utterly inhospitable to anything we would recognize as living. The silence of that ancient world was absolute. No bird sang. No insect chirped. No tree grew. There was only rock and water and sky, locked in an eternal, lifeless dance. Somewhere between that dead world and this living one, something extraordinary happened. Against all odds, in circumstances we are only beginning to understand, life emerged. Not fully formed, not complex, but in its most primitive, most basic form. A simple molecule that could make copies of itself. A tiny bubble of chemistry that could maintain itself against the chaos of the outside world. The first stirrings of biology on a planet that had known only physics and chemistry. This is the story of that emergence. It is a story written in rocks and chemicals, in the structure of our cells and the sequences of our genes. It is a story that spans billions of years, beginning in the deep darkness of the early ocean and ending with you, sitting here now, reading these words, a product of that first spark of life that has burned continuously for nearly 4 billion years. To understand how life began, we must travel back to a time before time was measured, to a world so alien it might as well be another planet. We must shrink ourselves down to the molecular level to see the dance of atoms and the formation of the first complex chemicals. We must think in timescales that dwarf human civilization, where 1,000,000 years is but a moment and 1,000,000,000 years is merely a chapter in a much longer story. The question of how life began has haunted humanity since we first gained the capacity to ask it. Ancient civilizations developed elaborate creation myths to explain the origin of living things. Gods molded humans from clay. Life sprang fully formed from the chaos of primordial waters. Supernatural forces breathed animation into dead matter. These stories satisfied the need for explanation, but they did not satisfy the need for understanding. As science developed, we began to probe this question with new tools and new methodologies. We learned that all life on Earth shares a common chemistry, a common cellular structure, a common genetic code. We discovered that the rocks beneath our feet contain a record of ancient life, fossilized remnants of creatures that lived and died hundreds of millions of years ago. We developed theories about how simple chemicals might have organized themselves into the first living systems. Yet even today, with all our advanced technology and accumulated knowledge, the origin of life remains one of the great unsolved mysteries of science. We have pieces of the puzzle, fragments of the story, but we do not yet have the complete picture. We know that life began. We know approximately when it began. We can even make educated guesses about where it began, but the precise mechanisms by which non-living chemistry became living biology remain tantalizingly out of reach. This documentary script will take you on a journey through what we do know, what we suspect, and what remains to be discovered. We will explore the conditions on the early Earth, the chemistry of life's building blocks, the leading theories about how these blocks came together, and the evidence that supports or challenges these theories. We will meet the scientists who have dedicated their careers to solving this puzzle, and we will peer into the laboratories where they are attempting to recreate the origin of life itself. Along the way, we will encounter concepts that challenge our intuition about what is possible. We will see how random chemical reactions, given enough time and the right conditions, can create order from chaos. We will learn how the laws of physics and chemistry, the same laws that govern the stars and the atoms, also govern the emergence of life. We will discover that the boundary between living and non-living is not as clear-cut as we might imagine. But more than anything, this is a story about transformation, about how the Earth itself changed from a lifeless rock to a living world, about how simple molecules became complex organisms, about how chemistry became biology, about how the universe became aware of itself, through us, through life. It is a story of emergence, of complexity arising from simplicity, of the extraordinary emerging from the ordinary. So settle in, relax, and prepare to journey back through the mists of deep time, to a world that no longer exists, to witness the greatest transition in the history of our planet. The journey from non-life to life. The origin of everything that we are and everything that we might become. Chapter 1. The ancient question, long before we developed the scientific method, long before we had microscopes or laboratories, or the ability to peer into the structure of matter itself, humans wondered about the origin of life. It is a question as old as consciousness itself. Where did we come from? Why are we here? What is the difference between the living and the dead? In the ancient world, these questions were answered by myth and religion. The Babylonians believed that the god Marduk created humans from the blood of a slain god mixed with clay. The ancient Egyptians believed that the god Khnum fashioned humans on a potter's wheel. The Greeks told of Prometheus molding humans from clay and Athena breathing life into them. In the Judeo-Christian tradition, God formed Adam from the dust of the ground and breathed into his nostrils the breath of life. These creation stories, while different in their details, shared common themes. Life came from non-life through the intervention of divine forces. Humans were special creations, separate from and superior to the animals. The living world was created in its current form, with each species fixed and unchanging. These ideas would dominate human thought for thousands of years. But not all ancient thinkers were satisfied with supernatural explanations. In ancient Greece, a tradition of natural philosophy emerged that sought to explain the world through observation and reason, rather than through appeals to the gods. These early philosophers, working without the benefit of modern scientific instruments or methods, nevertheless laid the groundwork for our current understanding of the natural world. Thales of Miletus, who lived around 600 BCE, proposed that water was the fundamental substance from which all things arose. Anaximander, his student, suggested that life arose from the interaction of heat and moisture, and that the first animals were born in moisture and gradually adapted to life on dry land. Empedocles proposed that the first living things were formed by the random combination of parts, with only those combinations that could survive and reproduce persisting into the future. These ideas, remarkably, anticipated some aspects of modern evolutionary theory. They recognized that life might have arisen through natural processes rather than supernatural intervention. They understood that the survival of living things depended on their ability to function in their environment. They grasped, in a rudimentary way, the concept of natural selection. But it was Aristotle, the great Greek philosopher and naturalist who lived in the 4th century BCE, who developed the most influential theory of the origin of life in the ancient world. Aristotle believed in what he called spontaneous generation, the idea that living things could arise spontaneously from non-living matter, provided that matter contained what he called pneuma, or vital heat. According to Aristotle, different forms of life required different degrees of vital heat. Simple animals like worms and insects could arise from decaying matter or moist earth. Mice could spring from dirty hay. Eels, which Aristotle never saw reproducing or bearing young, must arise spontaneously from the mud of rivers and ponds. Even some plants, he believed, could grow from the earth without seeds. This theory of spontaneous generation was not based on careful observation or experimentation. Aristotle saw maggots appearing on rotting meat and assumed they were generated by the meat itself. He saw mice in his grain stores and assumed they arose from the grain. He never observed the flies laying eggs on the meat or the mice giving birth to their young in hidden nests. Nevertheless, his theory seemed to explain what he observed, and his authority was such that his ideas would dominate Western thought for nearly 2,000 years. During the Middle Ages, Aristotle's works were translated into Latin and incorporated into Christian theology. Spontaneous generation became accepted doctrine, taught in universities and accepted by scholars throughout Europe. Indeed, elaborate recipes were developed for generating specific types of organisms through spontaneous generation. In the 17th century, the Flemish chemist Jan Baptist van Helmont provided a recipe for generating mice. Place a dirty shirt in a container with wheat grains, wait 21 days, and mice would appear. He claimed to have performed this experiment himself and observed the results. Other scholars provided recipes for generating wasps from dead donkeys, bees from dead bulls, and scorpions from basil leaves left to decompose in the sun. It seems absurd to us now that educated people could believe such things. But we must remember that these scholars lacked our modern understanding of reproduction, of germs, of the microscopic world. They saw what appeared to be spontaneous generation all around them, and without the tools to investigate more closely, they accepted the evidence of their eyes. The first cracks in the theory of spontaneous generation appeared in the 17th century with the work of Francesco Redi, an Italian physician and naturalist. Redi decided to test whether maggots really did arise spontaneously from rotting meat, or whether there might be another explanation. He set up a simple experiment. He placed meat in several jars, some covered with gauze and some left open to the air. In the open jars, flies landed on the meat and maggots soon appeared. But in the covered jars, no maggots developed, even though the meat rotted in the same way. Redi observed that flies laid eggs on the gauze covering the closed jars, and that maggots hatched from these eggs. He concluded that maggots did not arise spontaneously from the meat, but rather from eggs laid by flies. This was a revolutionary finding. For the first time, someone had used controlled experimentation to disprove spontaneous generation for a specific case. But Redi's work did not overturn the theory entirely. Many scientists argued that while larger animals like flies might not arise spontaneously, smaller organisms, particularly the microscopic creatures being discovered with the newly invented microscope, might still be generated spontaneously. The debate continued into the 19th century, when Louis Pasteur, the French chemist and microbiologist, designed a series of elegant experiments that finally put the theory of spontaneous generation to rest. Pasteur showed that microorganisms in air caused the spoilage of nutrient broths, not spontaneous generation from the broth itself. He designed special flasks with S-shaped necks that allowed air in but prevented microorganisms from reaching the broth. In these flasks, the broth remained sterile indefinitely. Pasteur's experiments demonstrated conclusively that life does not arise spontaneously from non-living matter under current conditions on Earth. All life, from the simplest bacteria to the largest animals, comes from pre-existing life. Life begets life, and only life. This principle, known as biogenesis, became one of the fundamental tenets of biology. But this created a paradox. If life can only come from life, how did the first life arise? The Earth had not always existed. Geologists had demonstrated that the planet was ancient, perhaps hundreds of millions or even billions of years old, and that it had changed dramatically over that time. There must have been a time before life existed. So how did life begin? Some scientists suggested that life might have come from space, carried to Earth on meteorites or comets. This theory, called panspermia, merely pushed the problem back a step. Even if life on Earth originated elsewhere, the question remained, how did it first arise? Others suggested that the origin of life was a unique event that could not be studied scientifically, that the conditions necessary for life's emergence no longer existed on Earth. It was not until the 20th century, with advances in chemistry, geology, and biology, that scientists began to develop testable theories about how life might have originated from non-living matter on the early Earth. These theories would require a deep understanding of the conditions on the young planet, the chemistry of organic molecules, and the fundamental requirements for life itself. Chapter 2. The Young Earth. To understand how life began, we must first understand the world in which it began. The Earth of 4 billion years ago was a radically different place from the Earth we know today. It was a world of fire and fury, of violent impacts and volcanic eruptions, of oceans that boiled and rocks that melted. It was a world that would kill any modern organism in moments. Yet somehow, in this hostile environment, life found a way to begin. The Earth formed approximately 5 billion years ago from a disk of gas and dust swirling around the young sun. Through a process called accretion, countless collisions between dust grains, rocks, and larger bodies gradually built up the mass of our planet. These impacts released tremendous amounts of energy, heating the growing Earth until its surface was molten. For millions of years, our planet was covered by an ocean of liquid rock, glowing red-hot and slowly cooling. This earliest period of Earth's history is called the Hadean Eon, named after Hades, the Greek underworld. It is an apt name. The Hadean Earth was a hellish place. The atmosphere, if it can be called that, was likely composed of hydrogen and helium blown in from the solar nebula, along with gases released by volcanic activity, water vapor, carbon dioxide, nitrogen, and possibly methane and ammonia. There was little or no oxygen in the air. The sky would have been a murky orange-brown, lit by the dull glow of the molten surface below. The young Earth was also subject to constant bombardment by asteroids and comets left over from the formation of the solar system. These impacts were far more frequent and violent than anything we experience today. Some of the impactors were large enough to vaporize oceans and melt the crust, essentially sterilizing the planet and requiring the entire process of cooling and ocean formation to begin again. The largest impact of this era created our moon. About 4 billion years ago, a Mars-sized body, sometimes called Theia, collided with the Earth in a cataclysmic impact that ejected a huge amount of material into orbit. This material eventually coalesced to form the moon. The impact liquefied the Earth's surface once again and may have stripped away much of the early atmosphere. It would take millions of years for the planet to cool and stabilize after this event. But gradually, inexorably, the Earth began to cool. As the bombardment decreased and the internal heat radiated away into space, the surface temperature dropped below the boiling point of water. Water vapor in the atmosphere began to condense, forming clouds. These clouds released their moisture as rain, and it rained, not for days or weeks, but potentially for millions of years, as the vast amounts of water vapor in the atmosphere condensed and fell to the surface. This rain collected in the low-lying areas of the cooling crust, forming the first oceans. These early oceans were nothing like the oceans we know today. They were hot, perhaps near boiling in some areas. They were acidic, filled with dissolved minerals and gases from volcanic emissions. They may have been a murky green or brown color, rather than the clear blue we associate with water. But they were liquid water, and liquid water would prove to be essential for the chemistry of life. The continents, if they existed at all during this time, were small and unstable. Tectonic activity was more vigorous than it is today due to the higher internal heat of the planet. Volcanic activity was widespread, constantly adding new gases to the atmosphere and new minerals to the oceans. Lightning storms, driven by the heat rising from the surface and the churning atmosphere, may have been near constant, crackling across the sky and striking the ground with tremendous energy. This was the world in which life would have to begin. A world of extremes, of violent change, of chemistry in constant flux. Yet despite its apparent hostility, this world possessed all the ingredients necessary for life. It had liquid water, the universal solvent in which biochemistry takes place. It had a diverse array of chemical elements brought together by the process of planetary formation. It had energy sources from the sun above and the volcanic heat below. And it had time, vast stretches of time in which random chemical reactions could explore every possible combination. The composition of the early atmosphere is a matter of some debate among scientists. For many years, it was believed that the atmosphere was strongly reducing, meaning it had little or no oxygen and was rich in hydrogen-containing gases like methane and ammonia. This composition would have been favorable for the synthesis of organic molecules, the building blocks of life. However, more recent studies suggest that the early atmosphere may have been less reducing than previously thought. Volcanic gases, which would have been a major source of atmospheric gases, tend to produce carbon dioxide, nitrogen, and water vapor with smaller amounts of hydrogen and other gases. Such a mildly reducing or even neutral atmosphere would make the synthesis of organic molecules more challenging, but not impossible. The early oceans were likely warm, perhaps 50 to 80 degrees Celsius in many regions, warmer than the modern oceans, but cool enough for complex chemistry to occur. They contained high concentrations of dissolved iron, giving them a greenish tint. They also contained dissolved carbon dioxide, making them acidic. Over time, chemical reactions between the water and the crustal rocks would have added other dissolved minerals: calcium, magnesium, sodium, chloride, and many others. The ocean floor was marked by volcanic vents, cracks in the crust where superheated water rich in dissolved minerals poured out into the surrounding ocean. These hydrothermal vents created localized regions of extreme chemistry, where hot, mineral-rich water mixed with cooler ocean water, precipitating minerals and creating chimney-like structures. These vents may have played a crucial role in the origin of life, as we will explore later. The sun, too, was different in the Earth's early days. The young sun was about 30% dimmer than it is today, producing less heat and light. Without a thick atmosphere to trap heat through the greenhouse effect, the Earth might have frozen solid. But the high levels of carbon dioxide and other greenhouse gases in the early atmosphere kept the planet warm enough for liquid water to exist. Ultraviolet radiation from the sun was intense, far stronger than it is today. Without an ozone layer to block this radiation, the surface of the Earth would have been bathed in damaging UV light. This radiation could break apart complex molecules and damage any emerging life forms. However, it could also provide energy for chemical reactions, driving the synthesis of organic molecules from simpler precursors. Tidal forces were also much stronger than they are today. The moon was closer to the Earth, and its gravitational pull created massive tides that swept across the shallow seas. These tidal cycles would have repeatedly wetted and dried coastal areas, concentrating organic molecules and potentially providing environments favorable for the development of early life. As the Hadean Eon gave way to the Archean Eon about 4 billion years ago, conditions on Earth became somewhat less extreme. The bombardment by large impactors decreased. The crust stabilized and thickened, forming the first stable landmasses. Volcanic activity continued but became less widespread. The oceans deepened and became less acidic as carbon dioxide was gradually absorbed into minerals in the crust. This is the world in which we believe life began. Not the calm, stable, hospitable Earth of later eons, but a dynamic, energetic, chemically rich environment where the boundary between the possible and the impossible was constantly being tested. A world where chemistry was king and where the right combination of circumstances might allow that chemistry to cross the threshold into biology. Chapter 3. The chemistry of life. Before we can understand how life began, we must first understand what life is, at least in chemical terms. When we strip away all the complexity and diversity of modern organisms, when we look past the cells and tissues and organs, we find that all life on Earth shares a common chemistry. We are all made from the same basic ingredients, organized in the same basic ways. Life, as we know it, is based on carbon. Carbon is the fourth most abundant element in the universe, after hydrogen, helium, and oxygen. But carbon is special. Unlike most elements, carbon atoms can form strong bonds not only with other elements but also with other carbon atoms, creating chains and rings and complex three-dimensional structures. This property, called catenation, allows carbon to form an almost infinite variety of molecules. Carbon can form 4 bonds with other atoms, giving it great versatility. It can bond with hydrogen to form hydrocarbons, the basis of oils and fats. It can bond with oxygen to form carbohydrates, the primary energy currency of life. It can bond with nitrogen to form amines and amino acids, the building blocks of proteins. It can form double bonds and triple bonds, creating molecules with different properties and reactivities. The molecules that make up living things are called organic molecules, not because they come from organisms, though they do, but because they are based on carbon. Organic chemistry is the study of carbon-containing compounds, and it is one of the richest and most complex branches of chemistry. There are millions of known organic compounds, and new ones are being discovered or synthesized every day. But while carbon is essential, it is not sufficient. Life also requires hydrogen, oxygen, nitrogen, phosphorus, and sulfur. These 6 elements, often abbreviated as CH-NOPs, make up about 98% of the mass of all living organisms. Each plays a specific role in the chemistry of life. Hydrogen is the simplest and most abundant element in the universe. In living things, it is usually bonded to carbon or oxygen. It is a component of water, the medium in which all biochemistry takes place. It is also part of all organic molecules and plays a crucial role in storing and releasing energy through chemical bonds. Oxygen is the third most abundant element in the universe and the most abundant element in the Earth's crust. It is highly reactive and forms bonds with most other elements. In living things, oxygen is found in water, in carbohydrates, in proteins, in nucleic acids, and in many other molecules. Many organisms use oxygen as the final acceptor of electrons in their energy-producing processes, though this was not always the case, and some organisms today still live without oxygen. Nitrogen is essential for the construction of amino acids and nucleotides, the building blocks of proteins and genetic material respectively. The atmosphere is about 78% nitrogen gas, but this molecular nitrogen is very stable and unreactive. Converting it into a form that can be used by living things requires significant energy, a process called nitrogen fixation. In the modern world, some bacteria and archaea can perform this process, and all other organisms depend on them for their supply of usable nitrogen. Phosphorus is found in nucleic acids, DNA and RNA, in ATP, the primary energy currency of cells, and in the phospholipids that make up cell membranes. Unlike the other major elements of life, phosphorus is relatively rare. It must be obtained from minerals in rocks and soil, and the availability of phosphorus often limits the growth of organisms in natural environments. Sulfur is found in some amino acids and plays important roles in protein structure and function. It is also involved in many metabolic processes and is essential for certain enzymes. Like phosphorus, sulfur must be obtained from the environment, though it is more widely available. From these 6 elements, life constructs 4 major classes of biological molecules: carbohydrates, lipids, proteins, and nucleic acids. Each class has specific functions and properties, and together they enable all the processes we associate with life. Carbohydrates are molecules made primarily of carbon, hydrogen, and oxygen, usually in a ratio of approximately 1:1. The simplest carbohydrates are sugars like glucose, which contains 6 carbon atoms arranged in a ring with hydrogen and oxygen atoms attached. Glucose and other simple sugars serve as fuel for cells, releasing energy when their bonds are broken. More complex carbohydrates, like starch and cellulose, are made by linking many sugar molecules together into long chains. Lipids, commonly called fats and oils, are diverse molecules that are generally hydrophobic, meaning they do not mix well with water. The most common lipids are fatty acids, long chains of carbon atoms with hydrogen atoms attached and an acidic group at one end. These fatty acids can be linked together in various ways to form fats, oils, waxes, and phospholipids. Phospholipids, with their water-loving head and water-fearing tail, are especially important because they spontaneously form the membranes that surround all cells. Proteins are large, complex molecules made from chains of smaller units called amino acids. There are 20 common amino acids used by life on Earth, each with a different structure and different properties. By linking these amino acids together in different sequences and lengths, cells can create millions of different proteins, each with a specific shape and function. Some proteins serve as structural components, giving cells their shape. Others act as enzymes, catalyzing the chemical reactions that keep cells alive. Still others transport molecules, send signals, or defend against disease. Nucleic acids are perhaps the most remarkable of all biological molecules. There are 2 main types: DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Both are made from long chains of units called nucleotides. Each nucleotide consists of a sugar, a phosphate group, and a nitrogen-containing base. There are 4 different bases in DNA: adenine, guanine, cytosine, and thymine, and 4 in RNA: adenine, guanine, cytosine, and uracil. DNA stores genetic information in the sequence of its bases. Like letters in an alphabet, the 4 bases can be arranged in any order, and different sequences encode different information. This information is used to build proteins, with each sequence of 3 bases, called a codon, specifying a particular amino acid. RNA plays multiple roles. It carries information from DNA to the protein-building machinery. It forms part of that machinery itself, and in some organisms, it serves as the genetic material. These 4 classes of molecules, working together in intricate networks of chemical reactions, make up what we call metabolism. Metabolism is the sum of all the chemical processes that occur in a living organism. It includes the breakdown of food molecules to release energy, the synthesis of new molecules needed for growth and repair, and the elimination of waste products. At the heart of metabolism is the concept of energy. Living things are not in equilibrium with their environment. They maintain concentrations of molecules, temperatures, and pH levels that differ from the surrounding world. Maintaining this disequilibrium requires energy. When organisms stop acquiring and using energy, they die and equilibrate with their surroundings. The energy for life comes ultimately from the sun, captured by photosynthetic organisms and converted into chemical energy stored in molecules like glucose. Other organisms obtain their energy by consuming these photosynthetic organisms, or by consuming organisms that have consumed them. Some organisms, particularly in the deep ocean, obtain energy from chemicals rather than from sunlight, using chemical reactions with minerals to power their metabolism. But energy alone is not enough for life. Life also requires information. Every living cell contains instructions for building and maintaining itself, instructions encoded in the sequence of nucleotides in its DNA. This information is copied when cells divide, ensuring that offspring resemble their parents. It is also expressed through the synthesis of proteins, with the information in genes directing the production of specific proteins that carry out specific functions. The relationship between nucleic acids and proteins is fundamental to life as we know it. Proteins do most of the work in cells, catalyzing reactions, building structures, and performing countless other tasks. But proteins cannot replicate themselves. They must be built according to instructions stored in nucleic acids. Yet nucleic acids cannot do anything on their own. They require proteins to copy them, to express their information, to repair damage. It is a chicken and egg problem. Which came first, the protein or the nucleic acid? This question is central to understanding the origin of life, and we will return to it shortly. But first, we must ask, where did the building blocks themselves come from? The amino acids, the nucleotides, the sugars, and lipids. Did they exist on the early Earth? Or did they have to be synthesized from simpler precursors? And if they had to be synthesized, what provided the energy and the conditions necessary for that synthesis? Chapter 4. The Building Blocks. In 1953, a young graduate student named Stanley Miller, working in the laboratory of Harold Urey at the University of Chicago, performed an experiment that would revolutionize our understanding of how life might have begun. Miller set up a closed system containing water, methane, ammonia, and hydrogen, gases thought to be present in the early Earth's atmosphere. He heated the water to produce steam, which mixed with the gases in a larger flask. Then he passed electric sparks through the mixture, simulating lightning strikes. After running the experiment for a week, Miller analyzed the contents of the flask. To his amazement, he found that the simple inorganic compounds had been transformed into a variety of organic molecules. Most remarkably, he had created several amino acids, the building blocks of proteins. This was the first demonstration that the basic molecules of life could be synthesized from simple inorganic precursors under plausible early Earth conditions. The Miller-Urey experiment, as it came to be known, captured the public imagination and energized the field of origin of life research. It showed that the synthesis of organic molecules was not a rare or unlikely event requiring special conditions or divine intervention. Rather, it was a natural consequence of the chemistry of carbon-containing compounds when supplied with energy. Since Miller's original experiment, scientists have performed thousands of similar experiments with different gas mixtures, different energy sources, and different conditions. These experiments have produced not just amino acids, but also nucleotide bases, sugars, and lipid precursors. They have shown that organic molecules can form under a wide range of conditions, not just those of Miller's original experiment. However, the Miller-Urey experiment and its successors have also revealed the challenges facing the prebiotic synthesis of life's building blocks. While amino acids form readily in many different conditions, other molecules are more difficult to produce. Sugars, in particular, are challenging. They form through a complex series of reactions and are unstable, breaking down or reacting with other molecules before they can accumulate in significant quantities. The synthesis of nucleotide bases is also problematic. While adenine, one of the 4 bases in DNA and RNA, can be synthesized relatively easily, the other bases are more challenging, and forming complete nucleotides, with the base attached to a sugar and a phosphate group, is even more difficult. The reactions tend to be inefficient, producing low yields and a complex mixture of products. Moreover, subsequent research has cast doubt on the assumption that the early Earth's atmosphere was strongly reducing, rich in methane and ammonia. Volcanic gases, likely the main source of atmospheric gases on the early Earth, tend to produce carbon dioxide, nitrogen, and water vapor rather than methane and ammonia. An atmosphere dominated by carbon dioxide would make the synthesis of organic molecules more difficult, though not impossible. This has led scientists to consider alternative sources for the organic molecules needed for life. One possibility is that they were delivered from space. Meteorites, the rocky debris left over from the formation of the solar system, are known to contain organic molecules, including amino acids. The Murchison meteorite, which fell in Australia in 1969, was found to contain more than 90 different amino acids, including many not found in living organisms. The presence of organic molecules in meteorites suggests that these molecules form naturally in space through chemical reactions in interstellar clouds or on the surfaces of asteroids. During the heavy bombardment period of the early Earth, countless meteorites and comets would have fallen to the surface, potentially delivering large quantities of organic molecules to the young planet. Comets, too, are rich in organic molecules. These icy bodies, formed in the outer solar system and occasionally perturbed into the inner solar system, contain water, carbon dioxide, methane, and more complex organic compounds. When they impact a planet, they can deliver both water and organic molecules. Some scientists have suggested that much of Earth's water, and perhaps much of its organic material, was delivered by comets. But whether formed on Earth or delivered from space, the question remains. How did these simple building blocks assemble into the complex molecules of life? Amino acids had to be linked together into proteins. Nucleotides had to be linked together into nucleic acids. Lipids had to organize into membranes. And all of these processes had to occur in the right place at the right time to produce the first living cell. One challenge is that the reactions that form biological polymers, as these long-chain molecules are called, generally require the removal of water. When amino acids link together to form a protein, a molecule of water is released. The same is true when nucleotides link to form nucleic acids. But on the early Earth, these reactions would have been taking place in water, in the oceans. How can a reaction that requires the removal of water proceed in an aqueous environment? Several solutions to this problem have been proposed. One is that the polymers formed not in the open ocean, but in specialized environments where water could be removed. Tidal pools, for instance, fill with seawater when the tide is high but dry out when the tide recedes. As the water evaporates, the concentration of dissolved molecules increases and polymerization reactions become more favorable. Another possibility is that polymerization occurred on the surfaces of minerals. Certain clay minerals, for instance, can adsorb organic molecules from solution, concentrating them on their surfaces. The surface of the mineral can also provide a template, organizing the molecules in ways that favor polymerization. Experiments have shown that amino acids and nucleotides can indeed polymerize on mineral surfaces under appropriate conditions. A third possibility involves the use of activating agents, molecules that make the polymerization reaction more favorable. In modern cells, ATP serves this role, providing the energy needed to drive the synthesis of proteins and nucleic acids. On the prebiotic Earth, other molecules might have served a similar function. Polyphosphates, for instance, are chains of phosphate groups that can donate energy to drive chemical reactions. But even if we can explain how biological polymers formed, we still face a fundamental problem. Not all amino acid sequences form functional proteins. Not all nucleotide sequences encode useful information. The space of possible sequences is vast, and the fraction of those sequences that are functional is vanishingly small. How did the first functional proteins and nucleic acids arise from random polymerization? This is where we encounter the central paradox of the origin of life. Modern cells use proteins to synthesize and copy nucleic acids, and they use the information in nucleic acids to build proteins. Each depends on the other, and neither can function alone. So which came first? How could a system evolve that required both proteins and nucleic acids when neither could exist without the other? For many years, this chicken and egg problem seemed insoluble. But in the 1980s, a discovery was made that would transform our understanding of early life and suggest a possible solution to the paradox. Scientists discovered that RNA, long thought to be merely a passive carrier of genetic information, could also act as an enzyme, catalyzing chemical reaction, as just like proteins do. These catalytic RNA molecules, called ribozymes, could perform many of the same functions as protein enzymes. They could cut and splice other RNA molecules. They could catalyze the synthesis of new RNA molecules. Most remarkably, some ribozymes could even make copies of themselves, or at least of short RNA sequences. This discovery suggested a possible solution to the chicken and egg problem. Perhaps RNA came first, serving both as genetic material and as catalyst. Chapter 5. The RNA world. The discovery of catalytic RNA fundamentally changed how scientists think about the origin of life. For the first time, it seemed possible that a single type of molecule could perform both of the essential functions required for life: storing genetic information and catalyzing chemical reactions. This led to the proposal of the RNA world hypothesis, the idea that life once existed in a form based entirely on RNA, without DNA or proteins. In the RNA world, as scientists envision it, RNA molecules served as the genes, encoding information about their own structure. At the same time, some RNA molecules folded into specific three-dimensional shapes that allowed them to catalyze chemical reactions, including the synthesis of more RNA. These catalytic RNAs could act on other RNA molecules, cutting them, joining them, and even copying them. In this way, a kind of metabolism based entirely on RNA could have existed. The RNA world hypothesis is attractive because it solves the chicken and egg problem. If RNA can act as both gene and enzyme, then a self-replicating system based on RNA could have evolved without requiring proteins or DNA. Later, DNA and proteins could have been added to the system, taking over the roles of information storage and catalysis respectively, because they perform these roles more efficiently than RNA. Indeed, there are good reasons to think that DNA and proteins are later refinements of an RNA-based system. DNA is more stable than RNA, making it better suited for long-term information storage. Proteins are more versatile than RNA as catalysts, able to perform a wider range of chemical reactions. But both DNA and proteins are synthesized using RNA as an intermediary, suggesting that RNA came first and the others were added later. Moreover, modern cells still retain what appear to be molecular fossils of the RNA world. The ribosome, the molecular machine that synthesizes proteins in all living cells, is made primarily of RNA. The catalytic activity that joins amino acids together to form proteins is performed by RNA, not protein. This suggests that protein synthesis evolved in an RNA world and that the RNA components of the ribosome are relics of that ancient system. Similarly, many of the key molecules involved in cellular metabolism are either RNA molecules or contain RNA-like components. ATP, the primary energy currency of cells, contains the same ribose sugar and adenine base found in RNA. Many vitamins and coenzymes, molecules that help enzymes function, also contain nucleotide-like components. This suggests that these molecules evolved in a world where RNA was the dominant biological molecule. But while the RNA world hypothesis is appealing in its simplicity, it also faces significant challenges. The biggest challenge is the difficulty of synthesizing RNA under prebiotic conditions. While the individual components of RNA, the nucleotide bases and the ribose sugar, can be synthesized in prebiotic chemistry experiments, linking them together to form nucleotides is difficult. And linking nucleotides together to form RNA is even more difficult. RNA is chemically unstable. In water, especially at high temperatures, RNA molecules break apart relatively quickly. The bonds linking the nucleotides together can be hydrolyzed, breaking the chain. The bases can detach from the sugar. The molecule can fold back on itself and catalyze its own destruction. All of these processes would have made it difficult for RNA to accumulate and persist on the early Earth. Moreover, RNA is a complex molecule. Each nucleotide contains 20 or more atoms arranged in a specific three-dimensional structure. The probability that such a structure would assemble spontaneously from simple precursors is extremely low. And even if nucleotides did form, they would have to polymerize in the correct way, linking together through specific chemical bonds to form functional RNA. Some scientists have proposed modifications to the RNA world hypothesis to address these challenges. One proposal is that RNA was not the first genetic molecule, but was preceded by simpler nucleic acid-like polymers that were easier to synthesize and more stable. These protonucleic acids could have served as the first genetic materials, later being replaced by RNA as the chemistry of life became more sophisticated. Another proposal is that the first self-replicating systems did not use nucleic acids at all, but were based on proteins or other types of molecules. Proteins can fold into complex shapes and catalyze chemical reactions, and some proteins can even catalyze the synthesis of other proteins under special conditions. Perhaps proteins came first, and nucleic acids were added later to provide a more efficient means of storing and transmitting information. A third proposal is that life began not with a single type of molecule, but with a complex mixture of molecules that work together. In this scenario, proteins, nucleic acids, and other molecules all played roles from the beginning, with the system as a whole exhibiting the properties of life even though no single component could replicate or catalyze reactions on its own. Despite these challenges and alternatives, the RNA world remains the leading hypothesis for the origin of life, primarily because of the central role that RNA plays in modern biochemistry and the ability of RNA to perform both genetic and catalytic functions. Scientists continue to work on demonstrating how RNA could have formed and replicated under prebiotic conditions. One of the key discoveries supporting the RNA world hypothesis is the existence of RNA molecules that can replicate themselves, at least partially. Several research groups have created ribozymes that can catalyze the synthesis of other RNA molecules, and some can even copy short sequences of RNA. While these ribozymes cannot yet copy sequences as long and complex as themselves, they demonstrate that self-replication by RNA is at least possible in principle. Another important discovery is that RNA molecules can evolve through a process analogous to natural selection. In a test tube containing many different RNA sequences and a supply of nucleotides, those sequences that can replicate most efficiently will increase in number, while less efficient sequences will decline. Over many generations of replication and selection, new and more efficient replicating sequences can evolve. This process, called in vitro evolution, shows that Darwinian selection can operate at the molecular level, even in the absence of cells or organisms. Scientists have used in vitro evolution to create RNA molecules with specific properties, ribozymes that bind to particular target molecules, that catalyze particular reactions, that respond to particular environmental conditions. These experiments show that RNA has the potential to evolve complex functions through natural selection, supporting the idea that RNA-based life could have evolved increasing complexity over time. But even if RNA could replicate and evolve, it still faces a critical limitation. It is exposed to the environment, vulnerable to degradation and dilution. In the open ocean, any successful replicating molecule would quickly diffuse away, its copies spreading out and becoming so dilute that they could no longer interact. The products of its catalytic activity would also diffuse away, benefiting other molecules rather than itself. What was needed was a way to keep related molecules together, to create a boundary between self and environment. This brings us to one of the most important inventions in the history of life: the cell membrane. A membrane is a barrier that separates the inside of a cell from the outside world, allowing the cell to maintain different chemical conditions on either side. In modern cells, membranes are made of phospholipids, molecules with a water-loving head and a water-fearing tail. When placed in water, these molecules spontaneously organize into double-layered sheets, with the water-loving heads facing the water on both sides and the water-fearing tails sequestered in the middle. These lipid bilayers can form closed spheres, called vesicles, that enclose a volume of water and any molecules dissolved in it. The vesicle creates a microenvironment separated from the surrounding solution, allowing different chemical conditions to be maintained inside and outside. Molecules produced inside the vesicle remain inside, rather than diffusing away into the environment. This concentration of molecules makes complex chemical reactions more likely and more efficient. The formation of vesicles from lipids is a spontaneous process driven by the hydrophobic effect. No enzymes or genetic information are required. Simply mixing certain lipids with water will produce vesicles automatically. This means that vesicles could have formed on the early Earth without requiring complex biological machinery, providing ready-made compartments in which the chemistry of life could develop. The combination of self-replicating RNA and lipid vesicles could have produced the first protocells, primitive cell-like structures that exhibited some of the properties of life. These protocells would have been far simpler than modern cells, lacking most of the complex machinery that modern cells possess. But they would have had the essential features: genetic information in the form of RNA, metabolism catalyzed by ribozymes, and a boundary, the lipid membrane, separating them from the environment. Such protocells could have undergone a form of evolution. Protocells containing more efficient self-replicating RNA would produce more copies of themselves. Protocells with membranes that allowed the right nutrients in and kept the products of metabolism inside would be more successful. Over time, natural selection would favor those protocells that were better at surviving and reproducing, leading to increasing complexity and efficiency. Chapter 6. The first cells imagine the early ocean, some 4 billion years ago. The water is warm, perhaps uncomfortably so by modern standards, and murky with dissolved minerals and suspended particles. The sky above is a dull orange-brown, thick with clouds that rarely part to reveal the sun. On the shore, volcanic vents belch steam and gases into the air. Lightning crackles across the sky, striking the water and the land with tremendous energy. Beneath the surface, invisible to any observer, a drama is unfolding at the molecular level. Amino acids and nucleotides, synthesized in the atmosphere or delivered by meteorites, dissolve in the water. They concentrate in tidal pools and on mineral surfaces. They link together into chains, forming primitive proteins and RNA molecules. Some of these molecules have catalytic activity, able to promote chemical reactions, including their own synthesis. Lipids, fatty acids synthesized from simpler precursors, also accumulate in the water. Being hydrophobic, they aggregate together, forming droplets and films on the water surface. Some of these lipid aggregates spontaneously form vesicles, tiny spheres of lipid bilayer enclosing a small volume of water. Most of these vesicles are empty, but some happen to enclose one or more RNA molecules. Inside these vesicles, a different chemistry becomes possible. The RNA molecules are no longer free to diffuse away into the vast ocean. They are concentrated in the tiny volume of the vesicle, where they are more likely to interact with each other and with other molecules. If one of these RNA molecules is a ribozyme that can catalyze the synthesis of more RNA, it can produce copies of itself, and these copies remain inside the vesicle rather than being lost to the environment. This is the beginning of a protocell, a primitive cell-like structure that exhibits some of the properties of life. It has a boundary, the lipid membrane, that separates it from the environment. It has genetic information encoded in the sequence of nucleotides in its RNA. And it has metabolism, the chemical reactions catalyzed by its ribozymes that allow it to extract resources from the environment and use them to make more of itself. Of course, these first protocells would have been incredibly simple compared to modern cells. They would have lacked most of the complex machinery that modern cells possess. No DNA, no proteins, no ribosomes, no ATP synthesis machinery, no transport proteins in their membranes. They would have been barely alive, teetering on the edge between chemistry and biology. But simple as they were, they could evolve. Different protocells would have had different RNA sequences, different compositions of lipids in their membranes, different internal chemistries. Those protocells that were better at surviving and reproducing would leave more descendants. Those that were less efficient would be outcompeted and disappear. Over countless generations, natural selection would refine and improve these protocells, leading to increasing complexity. One of the first challenges these protocells would have faced is obtaining the raw materials needed for growth and replication. Modern cells have elaborate systems for importing nutrients from the environment. Proteins embedded in the membrane that selectively transport specific molecules. But the first protocells would have had no such sophisticated machinery. How could they obtain nutrients from the environment? The answer may lie in the properties of primitive lipid membranes. Unlike modern cell membranes, which are highly selective about what they allow to pass, primitive membranes made of simple fatty acids are relatively leaky. Small molecules like nucleotides and amino acids can pass through these membranes relatively easily, while larger molecules like RNA and proteins cannot. This would allow protocells to absorb nutrients from the environment without requiring complex transport machinery. As these molecules entered the protocell, they could be incorporated into new RNA molecules or used in other chemical reactions catalyzed by ribozymes. The protocell would grow, its membrane expanding as new lipids were added. Eventually, the protocell might become unstable and divide into two smaller protocells, each containing some of the original RNA molecules. This simple form of reproduction would allow the protocell population to grow. But there is a problem with this scenario. If protocells simply absorbed nutrients from the environment and grew until they divided randomly, there would be no guarantee that each daughter cell would receive a complete set of the ribozymes needed for survival. Some daughter cells might get most or all of the ribozymes, while others might get few or none. The latter would not survive, representing a waste of resources. The solution to this problem is replication. Before the protocell divides, the RNA molecules inside must be copied so that there are enough copies to distribute to both daughter cells. In modern cells, this is accomplished by complex protein machinery that copies DNA with high fidelity. But in protocells, the copying would have been done by ribozymes, which are much less accurate and efficient. Nevertheless, some ribozymes discovered in modern laboratories can catalyze the synthesis of new RNA molecules using an existing RNA as a template. While these ribozymes cannot yet copy RNA molecules as long and complex as themselves, they demonstrate that RNA-based replication is possible. It is plausible that similar or more sophisticated ribozymes could have existed in ancient protocells, providing a means of copying genetic information. As protocells evolved, they would have faced increasing competition for resources. In the early days, when protocells were rare and nutrients were abundant, any protocell that could replicate would be successful. But as protocells became more numerous, they would have depleted the available supply of prebiotic organic molecules. Those protocells that could synthesize their own building blocks, rather than relying on the environment to provide them, would have had a significant advantage. This pressure would have driven the evolution of increasingly sophisticated metabolic pathways. Early ribozymes might have catalyzed simple modifications of existing molecules. Later ribozymes could catalyze more complex reactions, building larger molecules from smaller precursors. Eventually, networks of ribozymes working together could perform complex multi-step syntheses, constructing nucleotides and other essential molecules from simple starting materials. At the same time, protocells would have been evolving more sophisticated membranes. The simple fatty acid membranes of the earliest protocells were leaky and unstable, prone to breaking apart or fusing with other vesicles. The evolution of membranes made of more complex lipids, including phospholipids with different head groups and tail lengths, would have made protocells more stable and allowed better control over what entered and left the cell. Eventually, some protocells would have evolved the ability to synthesize proteins. Proteins are far more versatile than RNA as catalysts, able to perform a wider range of chemical reactions with greater efficiency. But proteins cannot replicate themselves or store genetic information. A system that used both RNA and proteins, with RNA storing information and proteins performing catalysis, would be more efficient than a system using RNA alone. The evolution of protein synthesis would have required the development of the genetic code, the correspondence between sequences of nucleotides in RNA and sequences of amino acids in proteins. Each sequence of 3 nucleotides, called a codon, specifies a particular amino acid. By stringing together different codons, different proteins with different sequences and different functions can be encoded. How the genetic code evolved is one of the great unsolved mysteries of biology. The code is nearly universal, used by all living organisms with only minor variations. This universality suggests that the code evolved very early, probably in the protocell stage, and was then inherited by all subsequent life. But how did it arise? Why do particular codons correspond to particular amino acids? We do not know, though many theories have been proposed. With the evolution of proteins came the evolution of enzymes, protein catalysts that could perform biochemical reactions with great efficiency and specificity. Enzymes could take over many of the roles previously performed by ribozymes, allowing the ribozymes to be lost or repurposed. The ribosome itself, the machine that synthesizes proteins, evolved during this period, though its catalytic core remains made of RNA, a relic of the RNA world. Another crucial innovation was the evolution of DNA. DNA is more stable than RNA, making it better suited for long-term information storage. It is also easier to repair when damaged. The evolution of DNA would have allowed cells to store larger genomes and maintain them more accurately over many generations of replication. DNA became the primary repository of genetic information, with RNA serving as an intermediary between DNA and proteins. With DNA, proteins, RNA, and sophisticated lipid membranes, protocells had become true cells, possessing all the essential features of modern life. They could store information in DNA, express that information through RNA and proteins, perform complex metabolic reactions to obtain energy and synthesize needed molecules, and reproduce themselves with reasonable fidelity. They had crossed the threshold from chemistry to biology. Chapter 7. The emergence of DNA and proteins. The transition from an RNA world to a world of DNA and proteins was one of the most important events in the history of life. It marked the transition from simple, inefficient protocells to sophisticated organisms capable of complex biochemistry and rapid evolution. But how did this transition occur? How did cells evolve the machinery to synthesize proteins and DNA when they had previously relied on RNA alone? The answer lies in the principle of evolution. Complex systems can evolve through a series of small, incremental changes, each of which provides some advantage to the organism. The evolution of protein synthesis and DNA did not happen all at once through a single miraculous mutation. Instead, it happened gradually through many small steps, each building on what came before. The first step toward protein synthesis may have been the evolution of amino acid-carrying ribozymes. In modern cells, transfer RNAs, tRNAs, carry amino acids to the ribosome, where they are assembled into proteins. Each RNA recognizes a specific codon in the messenger RNA and carries the corresponding amino acid. This system requires sophisticated molecular recognition, but it could have evolved from simpler precursors. Perhaps the earliest RNA-like molecules were simply RNA sequences that happened to bind to particular amino acids. Through natural selection, those RNA molecules that could bind amino acids and bring them into proximity with other amino acid-carrying RNAs might have facilitated the formation of peptide bonds, linking amino acids together. Over time, this system could have become more sophisticated, with specific RNAs recognizing specific codons and carrying specific amino acids. The ribosome itself, the molecular machine that synthesizes proteins in all modern cells, is primarily made of RNA. The protein components of the ribosome play supporting roles, but the actual catalysis of peptide bond formation is performed by ribosomal RNA. This strongly suggests that the ribosome evolved in the RNA world as an RNA-based machine for linking amino acids together. Early ribosomes would have been simpler and less accurate than modern ones. They might have made many mistakes, inserting the wrong amino acids or failing to recognize codons correctly. But even inefficient protein synthesis would have provided an advantage. Proteins, even randomly sequenced ones, might have had some catalytic activity or structural function, supplementing the ribozymes. Protocells with this ability would have outcompeted those without it. As protein synthesis became more efficient and accurate, proteins began to take over many of the catalytic roles previously performed by ribozymes. Proteins are more versatile than RNA, capable of performing a wider range of chemical reactions. They can fold into more complex and stable structures. They can incorporate metal ions and other cofactors that enhance their catalytic abilities. In nearly every way except replication and information storage, proteins outperform RNA. Over time, more and more ribozymes would have been replaced by protein enzymes. The RNA world gradually gave way to an RNA-protein world, where RNA stored genetic information and proteins performed most of the catalysis. Many ribozymes were lost entirely, their functions taken over by proteins. Others were retained because they performed functions that proteins could not easily replicate, or because they were integral to structures like the ribosome that had evolved early and were difficult to change. The evolution of DNA was similarly gradual. DNA differs from RNA in two main ways. It uses the sugar deoxyribose instead of ribose, and it uses the base thymine instead of uracil. Both of these changes make DNA more stable than RNA. Deoxyribose lacks a hydroxyl group that makes RNA prone to breaking apart. Thymine is methylated uracil, and this methylation makes DNA easier to repair when it is damaged. The first step toward DNA may have been the evolution of enzymes that could convert ribonucleotides into deoxyribonucleotides. Such enzymes exist in all modern cells, and they are essential for DNA synthesis. Once deoxyribonucleotides were available, they could be incorporated into nucleic acid strands alongside regular ribonucleotides. Strands containing a mixture of ribo and deoxyribonucleotides would have been more stable than pure RNA. Over time, cells evolved enzymes that could synthesize pure DNA using RNA as a template. These enzymes, called reverse transcriptases, exist in modern retroviruses and in some other organisms. They allow information to flow from RNA to DNA, the reverse of the usual direction. Once cells had the ability to copy RNA into DNA, they could store their genetic information in the more stable DNA while still using RNA for other purposes. The evolution of DNA as the primary genetic material would have provided several advantages. DNA is more stable than RNA, allowing cells to maintain larger genomes without accumulating too many errors. DNA is also double-stranded, with each strand serving as a template for the other. If one strand is damaged, the other can be used to repair it. This redundancy makes DNA much more reliable for long-term information storage. With DNA as the genetic material and proteins as the primary catalysts, cells had evolved the basic architecture that characterizes all modern life. Information flows from DNA to RNA to protein, a pattern so fundamental that it is often called the central dogma of molecular biology. DNA stores the information, RNA carries it to the ribosomes, and proteins carry out the instructions, performing the chemical reactions that keep the cell alive. But even with DNA, proteins, and sophisticated metabolism, these early cells were still very simple compared to modern organisms. They likely lived in extreme environments, perhaps in hot springs or around deep-sea hydrothermal vents, where the chemistry favorable for life was most intense. They had small genomes, encoding only the most essential functions. They reproduced slowly and inaccurately by modern standards. Nevertheless, they were alive. They could grow, reproduce, and evolve. And from these simple beginnings, all the diversity of life on Earth would eventually arise. Every plant, every animal, every fungus, every bacterium that has ever lived is descended from these first cells. We are all children of the RNA world, inheritors of the first successful experiment in biology. Chapter 8. The last universal common ancestor all life on Earth shares a common chemistry. We all use the same genetic code, with only minor variations. We all use the same 20 amino acids to build proteins. We all use ATP as our primary energy currency. We all have cells bounded by lipid membranes. These similarities are not coincidental. They point to a common origin, to a single ancestor from which all modern life descended. Scientists call this ancestor Luca, the last universal common ancestor. Luca was not the first living thing. It was not even necessarily the most successful organism of its time, but it was the ancestor of all life that survived to the present day. Every other lineage of early life, if they existed, went extinct without leaving descendants. We know nothing about them except what we can infer from the rocks and from the chemistry of modern cells. What was Luca like? We can never know for certain, because Luca lived billions of years ago and left no fossils that we can definitively identify. But we can make educated guesses by comparing modern organisms and looking for the features they all share. Those features that are universal, or nearly so, were likely present in Luca. Features that differ between organisms likely evolved after Luca, in the separate lineages that descended from it. Based on this comparative approach, scientists have reconstructed a tentative portrait of Luca. It was almost certainly a single-celled organism, probably similar in some ways to modern bacteria or archaea. It had DNA as its genetic material, stored in a circular chromosome. It had ribosomes to synthesize proteins, using the same genetic code used by all modern life. It had a lipid membrane to separate its internal chemistry from the external environment. Luca probably lived in a hot, chemically rich environment, perhaps around a hydrothermal vent on the ocean floor. It obtained energy not from sunlight, which requires complex photosynthetic machinery that likely evolved later, but from chemical reactions with minerals. It may have used hydrogen gas and carbon dioxide to produce methane and other organic molecules, a metabolism still used by some modern microbes called methanogens. Luca's genome was probably small, encoding only the most essential genes for survival and reproduction. It likely had genes for DNA replication, RNA transcription, protein synthesis, and basic metabolism. But it probably lacked many of the genes that modern organisms possess for dealing with oxygen, for photosynthesis, for complex regulation of gene expression, for movement, and sensing the environment. One of the most intriguing questions about Luca is when it lived. The oldest rocks on Earth that might contain evidence of life are about 3.8 to 4.1 billion years old. Some scientists claim to have found chemical signatures or microscopic structures in these rocks that could be evidence of life. But these claims are controversial. More definitive fossils of microorganisms appear in rocks about 3-5 billion years old. If Luca lived around 3-8 to 4 billion years ago, then life arose remarkably quickly after the Earth became habitable. The planet formed about 4-5 billion years ago and was too hot and too heavily bombarded by asteroids for life to survive for perhaps the first few 100 million years. This suggests that once conditions were right, life emerged fairly rapidly, at least on geological timescales. After Luca, life diversified into the three great domains that exist today: bacteria, archaea, and eukarya. Bacteria and archaea are both groups of single-celled microorganisms that lack a nucleus and other membrane-bound organelles. For many years, they were lumped together as prokaryotes and considered to be more closely related to each other than either was to eukarya, the domain that includes all complex life like plants, animals, and fungi. But molecular studies have revealed that archaea and eukarya are actually more closely related to each other than either is to bacteria. The tree of life has three main branches, with bacteria forming one branch and archaea and eukarya forming the other two branches of a later split. This means that Luca, the common ancestor of all three domains, lived before these splits occurred. The split between bacteria on one side and archaea plus eukarya on the other probably occurred very early in the history of life, perhaps within a few 100 million years of LUCA. The split between archaea and eukarya occurred later, though still billions of years ago. Each of these lineages then evolved independently, developing unique features and adaptations while retaining the core biochemistry inherited from Luca. Bacteria evolved to become incredibly diverse, occupying nearly every habitat on Earth. They developed photosynthesis, producing oxygen as a waste product and transforming the Earth's atmosphere. They evolved the ability to fix nitrogen, making this essential element available to other organisms. They learned to live in extreme environments, from the hottest hot springs to the coldest polar ice to the depths of the Earth's crust. Archaea, initially thought to be just unusual bacteria, turned out to be a completely separate domain of life with unique biochemistry. Many archaea live in extreme environments, such as hot springs, salt lakes, and oxygen-free sediments. They have unique lipids in their membranes, different from those of bacteria or eukaryotes. They also have unique enzymes and metabolic pathways. Eukarya, the domain that includes us, evolved larger and more complex cells with a nucleus and other membrane-bound organelles. Eukaryotic cells probably arose through a process called endosymbiosis, where one cell engulfed another, and the two entered into a permanent partnership. The mitochondria that provide energy to eukaryotic cells were once free-living bacteria. The chloroplasts that allow plants to photosynthesize were once free-living photosynthetic bacteria. But all of this diversification, all of this complexity, all of the incredible variety of life on Earth today descended from that one common ancestor, Luca. We are all, in a very real sense, siblings, sharing not just a common home on this planet, but a common origin billions of years ago. The bacteria in your gut, the trees outside your window, the insects in the garden, you yourself, are all family, all descended from that first successful experiment in biology. Chapter 9. Alternative theories. While the RNA world hypothesis and the protocell model are currently the most widely accepted ideas about how life began, they are not the only theories. Over the years, scientists have proposed many alternative mechanisms for the origin of life, each with its own strengths and weaknesses. Some of these alternatives challenge the RNA-first model directly, suggesting that other molecules or systems came first. Others propose different environments or different chemical pathways for the emergence of life. One alternative is the metabolism-first hypothesis. This theory proposes that before there were self-replicating molecules like RNA, there were self-sustaining networks of chemical reactions, primitive metabolism, that could maintain themselves and grow. These metabolic networks would not have required complex information-carrying molecules or enzymes. Instead, they would have been autocatalytic, with the products of one reaction catalyzing other reactions in the network. Proponents of the metabolism first hypothesis point out that even the simplest self-replicating RNA molecules are quite complex, with hundreds of nucleotides arranged in a specific sequence. The probability of such a molecule arising by chance is vanishingly small, but simple autocatalytic networks might arise more easily through the random combination of simple molecules. Once established, these networks could evolve and become more complex, eventually incorporating informational molecules like RNA. One version of the metabolism first hypothesis focuses on the role of minerals in catalyzing chemical reactions. Certain minerals, particularly iron-sulfur minerals found around hydrothermal vents, can catalyze many of the reactions of modern metabolism. Perhaps the first metabolisms were not based on organic enzymes at all, but on inorganic minerals. These mineral-catalyzed reaction networks could have provided the energy and the building blocks needed for the later evolution of RNA and proteins. Another alternative is the proteins first hypothesis. This theory suggests that proteins, not RNA, were the first biological molecules. Proteins can form more readily than RNA under some conditions, and they can fold into complex shapes that give them catalytic activity. Some proteins can even catalyze the synthesis of other proteins, suggesting a possible route to self-replication. However, the proteins first hypothesis faces a significant challenge. How can proteins store and transmit genetic information? Unlike nucleic acids, proteins do not have an obvious way to encode information that can be copied. Each protein is a linear chain of amino acids, but there is no natural mechanism for using one protein as a template to make an identical copy. Without a way to store and transmit information, protein-based life could not evolve in a Darwinian sense. Some researchers have proposed that the first proteins were not templated at all, but arose through random polymerization of amino acids. Protocells containing many different random protein sequences would have had different properties, and those protocells with useful proteins would have been more successful. But without a way to reliably reproduce those useful proteins, this advantage would be lost when the protocell divided. A third alternative is the lipid world hypothesis. This theory suggests that self-replicating lipid vesicles came first, before RNA or proteins. Lipid vesicles can grow by absorbing more lipids from the environment, and they can divide when they become too large, creating two smaller vesicles. If the vesicles happen to contain some catalytic molecules that could promote the synthesis of more lipids, they could exhibit a kind of primitive metabolism and reproduction. The lipid world hypothesis is attractive because lipid vesicles form spontaneously and can evolve through natural selection, even without genetic information. Vesicles with different lipid compositions would have different properties, different stabilities, different growth rates. Those vesicles that were better at surviving and dividing would become more numerous. Later, RNA or other informational molecules could have been incorporated into the vesicles, adding genetic information to the system. Yet another alternative is panspermia, the idea that life did not originate on Earth at all, but was brought here from elsewhere in space. Microorganisms or the precursors of life could have traveled through space on meteorites or comets, eventually landing on the young Earth and seeding the planet with life. This idea is not as far-fetched as it might seem. We know that meteorites from Mars have landed on Earth, and it is possible that they could carry microorganisms if any existed on Mars. Some bacteria can survive for long periods in the vacuum and radiation of space. Experiments have shown that certain microbes can endure conditions similar to those on the surface of a meteorite during its journey through space. If microbial life existed elsewhere in the solar system, it is at least conceivable that it could have been transferred to Earth through impact events. However, panspermia does not really solve the problem of life's origin. It merely moves the problem to a different location. Even if life on Earth came from space, the question remains: how did that life originate in the first place? Where in the universe did the first living organism arise, and under what conditions? For this reason, most scientists view panspermia as a secondary question. The primary question is still how life arose from non-living matter, whether on Earth or elsewhere. A related idea is directed panspermia, the hypothesis that life was deliberately sent to Earth by an intelligent extraterrestrial civilization. This idea, while interesting as science fiction, is not considered a serious scientific hypothesis because it requires the existence of advanced aliens and provides no testable predictions. Like panspermia in general, it also does not explain how life originally arose. It just pushes the question back to the origin of the alien civilization. All of these alternative theories have their merits and their challenges. The RNA world hypothesis remains the leading theory because it provides a plausible solution to the chicken and egg problem of genes and enzymes, and because RNA plays such a central role in modern biochemistry. But the origin of life is still an active area of research, and it is possible that future discoveries will favor a different model or reveal that the truth combines elements of several different theories. What is clear is that the origin of life is a complex problem that will require input from many different fields of science: chemistry, biology, geology, astronomy, physics. It will require clever experiments to test different hypotheses and careful observation of the natural world to uncover clues about how life might have begun. And it will require an open mind, willing to follow the evidence wherever it leads, even if that means abandoning cherished theories in favor of new ones. Conclusion: the continuing mystery. And so we come to the end of our journey through deep time, back to a world that no longer exists, to witness the origin of life on Earth. We have seen how the young planet, hot and hostile, gradually cooled to create conditions where life could begin. We have explored the chemistry of life, the building blocks of amino acids and nucleotides, the organic molecules that make biology possible. We have encountered the RNA world, where a single type of molecule could both store information and catalyze reactions, providing a plausible solution to the chicken and egg problem of genes and enzymes. We have seen how lipid vesicles could have provided the compartments needed to concentrate organic molecules and create the first protocells. We have traced the evolution of these protocells into true cells with DNA, proteins, and sophisticated metabolism. We have met Luca, the last universal common ancestor, from whom all modern life descended. And we have considered alternative theories, different ideas about how life might have begun, each offering a different perspective on this fundamental question. Yet for all that we have learned, the origin of life remains a mystery. We have pieces of the puzzle, fragments of the story, but we do not yet have the complete picture. We know that life began, we know approximately when it began, we can make educated guesses about where and how it began, but we do not yet understand all the details. The precise sequence of events that led from simple organic molecules to the first living cell remains unknown. But this is not a cause for despair. Rather, it is an invitation to continued investigation, to further research, to new discoveries. Every year, scientists learn more about the chemistry of the early Earth, about the capabilities of RNA and other molecules, about the environments where life might have begun. Every year, we get a little closer to understanding this profound transition from chemistry to biology. In laboratories around the world, scientists are trying to create life from scratch, to recreate the conditions of the early Earth and see if life will emerge. They are synthesizing RNA molecules that can replicate themselves, creating protocells that can grow and divide, discovering new ribozymes with new capabilities. While no one has yet created a truly living cell from non-living components, the progress has been remarkable. We are also learning from the study of extreme environments on modern Earth. Microorganisms have been found in hot springs, in acidic lakes, in deep-sea hydrothermal vents, in Antarctic ice, even miles underground in solid rock. Each of these environments provides clues about the range of conditions where life can survive and potentially where it might originate. Some of these modern extremophiles may be living in environments similar to those where the first life arose. The search for life elsewhere in the universe also informs our understanding of life's origin. If we discover life on Mars, on the moons of Jupiter or Saturn, or on planets orbiting other stars, it will tell us whether life is common or rare in the universe. If that life uses different chemistry from Earth life, it will tell us whether our particular biochemistry is inevitable or just one of many possibilities. But beyond the scientific questions, the origin of life touches on deeper philosophical and existential issues. It speaks to who we are and where we came from. It tells us that we are not separate from nature but part of it, that we are made of the same atoms and molecules as everything else in the universe. It reveals that life is not a supernatural gift but a natural process, arising from the laws of chemistry and physics. It also tells us that we are connected, in the most fundamental way, to every other living thing on this planet. The tree in your yard, the bird in the sky, the bacteria in your gut, you yourself, are all descended from that first protocell that emerged in the ancient ocean. We all share the same genetic code, the same biochemistry, the same molecular machinery. We are all family, all children of Luca, all participants in the grand experiment of life on Earth. This connection should fill us with wonder and with responsibility. Wonder at the incredible journey that life has taken, from simple molecules to complex organisms, from a single cell to the diversity of the modern world. Responsibility to protect and preserve this legacy, to ensure that the experiment continues, that the chain of life that began nearly 4 billion years ago does not end with us. The origin of life is one of the great stories of our universe, a story of transformation and emergence, of order arising from chaos, of the extraordinary emerging from the ordinary. It is a story that began in the darkness of the early ocean and continues today in every living thing. It is our story, the story of how we came to be. As you drift off to sleep tonight, consider this. Every atom in your body was forged in the heart of a star. Every molecule in your cells was assembled through billions of years of evolution. You are a product of the universe becoming aware of itself, a temporary arrangement of matter and energy that can think and feel and wonder. You are a part of the great story of life on Earth, a story that began with simple chemistry and may someday reach the stars. The question of how life started on Earth is not just a scientific puzzle to be solved. It is a window into the nature of reality itself, into the creative power of the universe, into the potential for matter to organize itself into ever more complex forms. It reminds us that we live in a universe where the impossible becomes possible, where dead matter can become living, where consciousness can emerge from chemistry. So rest easy, knowing that you are part of something larger than yourself, part of an unbroken chain of life stretching back billions of years to the first cell, the first protocell, self-replicating molecule in the ancient ocean. You are a child of the stars, a product of deep time, a miracle of chemistry and biology. You are alive, and in being alive, you are connected to everything that has ever lived and everything that will ever live. Sleep well and dream of ancient oceans, of lightning striking primordial soup, of RNA molecules dancing in lipid vesicles, of the first cell dividing in the warm waters of the early Earth. Dream of the great story of life, and know that you are part of it. Good night. Chapter 10. Where Life Began. The search for the cradles of life. One of the most fascinating questions in origin of life research is not just how life began, but where it began. The early Earth offered many different environments, each with its own unique chemistry and energy sources. Some of these environments seem more promising than others for the emergence of life, and scientists have focused their attention on several key candidates. For many years, the dominant idea was that life began in shallow surface waters, in tide pools or lakes where organic molecules could concentrate through evaporation cycles. Lightning and ultraviolet radiation could provide energy for chemical reactions. However, this surface scenario has problems. The early Earth was heavily bombarded by asteroids, and the surface lacked protection from harmful UV radiation. These factors make surface origins less likely. This led scientists to consider deep-sea hydrothermal vents. When these vents were discovered in 1977, surrounded by thriving ecosystems in complete darkness, it revolutionized thinking about where life could exist. The vents provide chemical energy, protection from impacts and radiation, and mineral surfaces that could catalyze reactions. Different types of vents offer different conditions, with alkaline vents like those at Lost City being particularly promising. Alkaline hydrothermal vents produce warm fluids rich in hydrogen gas, creating chemical gradients similar to those modern cells use for energy. The porous mineral structures provide compartments where organic molecules could concentrate. Iron-sulfur minerals at these vents can catalyze reactions similar to modern metabolism. These features make alkaline vents an attractive candidate for the birthplace of life. However, other environments remain viable candidates. Some scientists favor volcanic rocks on land, where geothermal activity provides energy. Others suggest ice environments, where organic molecules concentrate in liquid veins. Each scenario has supporters and detractors, and the truth may never be known with certainty. Perhaps life attempted to emerge in multiple locations, with only one or a few successes. The diversity of possible environments reminds us that the early Earth was a vast natural laboratory, conducting countless experiments in chemistry across millions of years. Most experiments failed, but at least one succeeded spectacularly, giving rise to all the life we see today. Chapter 11. The Evidence. Reading the Ancient Record. How do we know anything about life's origin? An event that occurred billions of years ago? The answer lies in multiple lines of evidence from ancient rocks, modern molecular biology, and laboratory experiments. Together, these provide a fragmentary but increasingly clear picture of how life emerged. The most direct evidence would be fossils, but finding fossils of microscopic organisms from 4 billion years ago is extraordinarily difficult. The oldest rocks have been heavily altered by heat and pressure. Nevertheless, potential evidence exists in 3 5-billion-year-old stromatolites from Australia and even older rocks from Greenland that may contain traces of early life. Chemical signatures provide another window into ancient life. Living organisms preferentially use lighter isotopes of carbon, leaving a characteristic signature. Carbon isotope ratios in ancient rocks suggest that life may have existed as early as 3.8 to 4.1 billion years ago, though these interpretations remain controversial. Modern molecular biology offers insights through comparative genomics. By comparing the genes of different organisms, scientists can reconstruct what their common ancestor Luca was like. These studies suggest Luca lived 3.5 to 3.9 billion years ago and already possessed sophisticated cellular machinery, meaning simpler precursors must have existed even earlier. Laboratory experiments test hypotheses about prebiotic chemistry. While no experiment has yet created life from scratch, researchers have demonstrated that life's building blocks can form naturally, that RNA can act as both gene and enzyme, and that simple self-replicating systems are possible. Each experiment adds another piece to the puzzle. The search for life elsewhere in the solar system and beyond also informs our understanding. If life arose on Mars or exists in the oceans of Europa or Enceladus, It would tell us that the origin of life is not a rare fluke, but a common occurrence when conditions are right. Chapter 12. The Timeline. Mapping the journey from chemistry to biology. Let us consider the timeline of events that led from the formation of Earth to the emergence of the last common ancestor of all life. This timeline is necessarily speculative, based on fragmentary evidence, but it provides a framework for understanding the sequence of innovations. 56 billion years ago, Earth forms from material in the solar nebula. The young planet is molten and constantly bombarded. 5 billion years ago, the moon-forming giant impact occurs, liquefying Earth's surface again. 4 billion years ago, the surface solidifies and water begins to rain down, forming the first oceans. 1 to 3. 9 billion years ago marks the late heavy bombardment, when massive impacts repeatedly sterilized the surface. Yet somewhere around 0 billion years ago, in protected environments like deep-sea vents, organic chemistry begins to become more complex. Prebiotic molecules accumulate and undergo increasingly sophisticated reactions. Between 0 and 8 billion years ago, the first protocells emerge. Lipid vesicles encapsulate collections of self-replicating molecules. The RNA world flourishes, with ribozymes performing both genetic and catalytic functions. Natural selection begins operating at the molecular level. From 8 to 5 billion years ago, the transition to DNA-RNA protein world occurs. Proteins replace most ribozymes as catalysts. DNA becomes the primary genetic material. True cells with sophisticated metabolism emerge. By 6 billion years ago, Luca, the last universal common ancestor, is living and thriving. By 5 billion years ago, definitive microbial fossils appear in rocks. Life has successfully established itself and will never again be entirely absent from Earth. This timeline spans over 500 million years, longer than the entire Phanerozoic eon. The origin of life was not a sudden event but a gradual process unfolding across immense stretches of time. Epilogue. Reflections on our origins. As we return from our journey through deep time, we carry with us profound insights about existence and our place in the cosmos. The story of life's origin is more than scientific history. It speaks to fundamental questions about meaning, connection, and possibility. We are not separate from nature, but integral to it. The atoms in your body were forged in ancient stars. The molecules in your cells were refined through 4 billion years of evolution. You are made of stardust and ancient oceans, a temporary arrangement of matter that can think and feel and wonder about its own origins. Life's origin reveals the universe's inherent creativity. Given the right conditions, matter does not sit inertly but explores possibilities, builds complexity, creates novelty. You are evidence of this creativity, a product of natural laws operating over cosmic timescales. In you, the universe has become aware of itself. The story emphasizes both contingency and inevitability. Chance played a role, countless lucky breaks allowed life to emerge and persist. Yet given similar conditions elsewhere, life may arise again. The same chemistry operates throughout the cosmos. We may not be alone. As you rest tonight, remember that you are alive, maintaining yourself against entropy, participating in the oldest continuous process on Earth. You are a link in an unbroken chain stretching back 4 billion years to that first protocell in an ancient ocean. You are stardust contemplating stars, life contemplating life, the universe aware of itself. Sleep well, child of the stars. You are part of the greatest story ever told, a story that began with the Big Bang and continues through you. In your awareness lies something sacred, something profound, something worth celebrating. May your dreams be filled with ancient oceans and the first dawn of life. Good night. Chapter 13. Molecular Evolution. The dance of molecules over deep time. To truly understand how life began, we must think about evolution at the molecular level. Long before there were cells or organisms that we would recognize as alive. Molecular evolution is the process by which molecules change over time through variation and selection. It operates according to the same basic principles as biological evolution, but on a much smaller, more fundamental scale. In the prebiotic world, molecules were constantly forming and breaking apart. Most lasted only moments before degrading or reacting with something else. But occasionally, a molecule would form that was more stable than average, that resisted breakdown, that could persist for longer periods. These stable molecules accumulated, becoming more common in the chemical mixture. This was the first form of selection, not biological but chemical, favoring molecules that could survive. As the concentration of organic molecules increased, something more interesting became possible. Some molecules could catalyze reactions that produced more molecules like themselves. These autocatalytic molecules could amplify themselves, creating positive feedback loops. A single catalytic molecule could produce thousands of copies of itself, spreading through the chemical mixture like a chain reaction. This was a crucial step toward life, the ability of molecular patterns to reproduce themselves. But autocatalysis alone is not quite replication. True replication requires not just making more of something, but making accurate copies that preserve information. This is where template-directed synthesis becomes important. In template-directed synthesis, a molecule serves as a pattern for making another molecule with a complementary structure. DNA replication uses this principle, with each strand serving as a template for synthesizing a new complementary strand. RNA can also undergo template-directed synthesis. In the right conditions, with the right building blocks and energy sources, an RNA strand can serve as a template for making a copy of itself. The sequence of bases in the original strand determines the sequence in the copy through base pairing rules. This allows information encoded in the sequence to be preserved and passed on, a fundamental requirement for evolution. Once you have replicating molecules, you have the raw material for natural selection. Different molecular sequences will replicate at different rates. Some will be copied more accurately than others. Some will be more stable and survive longer. Those that replicate faster, more accurately, or survive longer will increase in number relative to others. Over time, the population of molecules will evolve, becoming better at replication and survival. This process has been demonstrated in the laboratory. Scientists have created systems of self-replicating RNA molecules and subjected them to selection for various properties. Speed of replication, ability to function in different chemical environments, resistance to degradation. In every case, the RNA evolves, with new variants arising and spreading if they have advantages. It is evolution in action, at the molecular level, demonstrating that Darwinian principles operate even in simple chemical systems. One of the most fascinating experiments in molecular evolution was conducted by Gerald Joyce and his colleagues. They created a system of two RNA molecules that could replicate each other through cross-catalysis. Each molecule acted as an enzyme that synthesized the other from building blocks in the solution. Together, they formed a minimal self-replicating system. When subjected to selection pressure, such as changes in temperature or salt concentration, the system evolved, with mutations arising and spreading if they improved replication under the new conditions. These experiments show that the key ingredients for evolution are remarkably simple. You need variation, differences between individuals in a population. You need heredity, the ability of offspring to resemble parents. And you need selection, differential survival or reproduction based on traits. These three ingredients are sufficient for evolution to occur, whether in molecules, cells, or complex organisms. In the prebiotic world, these ingredients would have been present. Random chemical synthesis and spontaneous mutations would create variation in molecular sequences. Template-directed replication would provide heredity, and differential stability and replication rates would provide selection. Given these conditions and enough time, molecular evolution would inevitably occur, generating increasingly complex and sophisticated replicating systems. But molecular evolution faces challenges that biological evolution does not. Molecules do not have cell walls to protect them from degradation. They do not have metabolisms to repair damage or generate the building blocks they need. They float freely in solution, where they can be diluted, degraded, or washed away. For molecular evolution to lead to life, some way of compartmentalizing and protecting successful molecular systems was needed. This is where lipid vesicles become crucial. By encapsulating replicating molecules within lipid membranes, protocells could protect their molecular systems while still allowing exchange with the environment. Molecules that improved replication or metabolism would benefit the protocell containing them, creating a link between molecular function and cellular success. This allowed molecular and cellular evolution to proceed in parallel, each driving improvements in the other. Chapter 14. Life and the Laws of Physics. How complexity arises from chaos. One of the most profound questions about life's origin is how it squares with the second law of thermodynamics. This law states that entropy, a measure of disorder, always increases in a closed system. Order naturally tends toward disorder. Hot things cool down. Organized things fall apart. Yet life is highly organized, maintaining complex structures and resisting the slide toward equilibrium. How can life exist in a universe governed by the second law? The answer lies in understanding that the Earth is not a closed system. Energy flows in from the sun and radiates back out into space. This flow of energy allows local decreases in entropy, local increases in organization, as long as the overall entropy of the universe increases. Life is one such local decrease, a pocket of organization sustained by the flow of energy through it. Think of a whirlpool in a stream. The whirlpool is a structured, organized pattern, distinct from the chaotic flow of water around it. Yet it does not violate any laws of physics. It exists because energy is flowing through the system, from upstream to downstream. The whirlpool is dissipating that energy, converting the organized flow of water into turbulent eddies and eventually heat. As long as the stream flows, the whirlpool persists. Life is similar. Living organisms are patterns of organization that persist by dissipating energy. We take in food or sunlight, extract the energy, use it to maintain our structures and functions, and release waste heat. As long as the energy keeps flowing, we persist. When it stops, we die and equilibrate with our surroundings, our complex organization dissolving back into simpler chemicals. This perspective helps us understand how life could emerge from non-living matter. Given a flow of energy through a chemical system, local increases in complexity and organization become not just possible, but probable. The system will naturally explore different configurations, and those that are good at capturing and dissipating energy will tend to persist and amplify. Over time, increasingly complex energy-dissipating structures can emerge. The early Earth was awash in energy flows. Sunlight streamed down from above. Heat flowed up from below through volcanic vents and geothermal systems. Chemical disequilibria existed between different environments, between vent fluids and seawater, between oxidized and reduced chemicals. These energy flows provided the power for chemical reactions, driving the synthesis of complex organic molecules. In this energetic environment, any molecular system that could capture and channel energy would have an advantage. Autocatalytic cycles that used energy to produce more of themselves would grow and spread. Replicating molecules that could harness energy flows would outcompete those that could not. Gradually, through countless cycles of variation and selection, energy channeling molecular systems would become more sophisticated and efficient. This thermodynamic perspective explains why life seems so well adapted to extracting energy from its environment. It did not start with metabolism and then later evolved the ability to extract energy. Rather, the ability to extract and dissipate energy was fundamental from the beginning. Life is, in essence, a particularly sophisticated way of dissipating energy gradients, one that maintains complex structures in the process. Some scientists have proposed that the emergence of life was not an unlikely accident, but a thermodynamic inevitability. Given the right conditions and energy flows, complex self-organizing systems will naturally emerge as the most efficient way to dissipate energy. Life is what chemistry does when trying to reach equilibrium in the presence of strong energy flows. By this view, life is not fighting against the second law but expressing it, organizing locally to dissipate energy and increase entropy globally. This idea remains controversial, but it provides a powerful framework for thinking about life's origin. It suggests that life is not a cosmic fluke but a natural outcome of physical law. Wherever similar conditions exist, with liquid water, energy flows, and organic chemistry, life may emerge. The universe may be full of life, all arising through the same thermodynamic principles that govern stars and weather patterns. Chapter 15. The Frontier. What we still do not know despite decades of research and remarkable progress. Many fundamental questions about the origin of life remain unanswered. These open questions represent the frontier of origin of life research, the challenges that will occupy scientists for years or decades to come. Understanding what we do not know is as important as understanding what we do know, for it guides future research and keeps us humble about our current theories. One major question is how RNA first formed on the early Earth. While we can synthesize RNA building blocks in the laboratory, getting them to link together into long chains remains challenging. RNA is unstable in water, especially at high temperatures. The chemistry that forms RNA seems to work best in conditions very different from what we think the early Earth was like. Did RNA form through currently unknown chemical pathways? Or was there a simpler precursor that later evolved into RNA? Another question concerns the origin of homochirality. Biological molecules like amino acids and sugars come in two mirror image forms, left-handed and right-handed. Life uses exclusively left-handed amino acids and right-handed sugars. Why? And how did this uniformity arise? In prebiotic chemistry, both forms would have been produced in equal amounts. Breaking this symmetry and selecting for one form over the other is a puzzle that remains unsolved. The origin of the genetic code poses another mystery. Why do particular 3 base codons correspond to particular amino acids? The code seems arbitrary, with no obvious chemical reason why one codon should specify one amino acid rather than another. Yet the code is nearly universal, used by all living things with only minor variations. This suggests it evolved very early, probably in the protocell stage, but how and why remain unclear. The transition from geochemistry to biochemistry is poorly understood. Modern metabolism uses enzymes that are themselves products of metabolism, creating a bootstrapping problem. How did the first metabolic pathways arise without enzymes? Could minerals have served as primitive catalysts, later replaced by protein enzymes? Or did metabolism emerge in a completely different way that we have not yet imagined? The origin of cellular compartmentalization raises questions. While lipid vesicles form spontaneously, incorporating complex molecular machinery into these vesicles while maintaining their integrity is non-trivial. How did the first protocells manage to grow and divide while keeping their internal chemistry separated from the environment? How did they evolve the sophisticated membrane proteins that modern cells use to control what enters and leaves? The location where life began remains uncertain. While deep-sea alkaline vents are currently favored by many researchers, other environments remain possible. Did life begin in one specific location and spread from there, or did it emerge independently in multiple locations? If the latter, why did only one lineage survive? Or did multiple lineages merge, combining different innovations developed in different locations? The timeline of life's origin is poorly constrained. We know life existed by 3.5 billion years ago, and possibly as early as 3, 8, or even 4.1 billion years ago. But exactly when did the transition from chemistry to biology occur? How long did it take? Was it a gradual process spanning hundreds of millions of years, or did it happen relatively quickly once certain thresholds were crossed? Perhaps the biggest question is whether we can recreate the origin of life in the laboratory. No one has yet created a truly living cell from non-living chemicals. We have made progress on pieces of the puzzle, creating self-replicating molecules, self-assembling vesicles, and simplified metabolic networks. But integrating these pieces into a coherent, evolving, growing system remains elusive. Will we eventually succeed, or are we missing some crucial insight? These questions remind us that, for all our progress, the origin of life remains one of the great unsolved mysteries of science. We have theories and hypotheses, some better supported than others, but we do not yet have a complete, detailed, universally accepted account of how life began. This is not a failure but an opportunity. It means there is still fundamental knowledge to be discovered, still revolutionary insights waiting to be found. The answers to these questions will likely come from multiple disciplines working together. Chemists will explore new prebiotic reactions. Biologists will study the simplest living organisms for clues about ancient life. Geologists will search for evidence in ancient rocks. Astronomers will look for life elsewhere. Physicists will develop new theories about self-organization and complexity. Computer scientists will model the origin of life in simulations. Together, these efforts will gradually illuminate the darkness, bringing us closer to understanding our ultimate origins. Chapter 16, Laboratory Adventures, Recreating the Origin of Life. Throughout this journey, we have discussed theories and hypotheses about how life might have begun. But science is not just about ideas. It is about testing those ideas through experiments and observations. For decades, scientists have been attempting to recreate aspects of life's origin in their laboratories, conducting experiments that simulate the conditions of the early Earth and observing what chemical reactions occur. These experiments have yielded remarkable insights and brought us closer to understanding how life could have emerged from non-living matter. The story of experimental origin of life research begins with Stanley Miller and Harold Urey in 1952. Miller was a graduate student at the University of Chicago, working under the supervision of Urey, a Nobel Prize-winning chemist. They designed an experiment to test whether organic molecules, the building blocks of life, could form from simple inorganic gases under conditions thought to resemble the early Earth atmosphere. Their experimental setup was elegantly simple. A glass apparatus contained water, representing the primordial ocean, which was heated to produce water vapor. This vapor mixed with a gas mixture of methane, ammonia, and hydrogen in a larger flask, representing the atmosphere. Electrodes passed through this flask, producing electric sparks to simulate lightning. The gases then flowed through a condenser where they cooled and dissolved back into water, completing the cycle. The system ran continuously for a week, with the water circulating through the apparatus over and over. When Miller analyzed the water after a week, the results exceeded all expectations. The once clear water had turned yellowish-brown, indicating that complex chemical reactions had occurred. Chemical analysis revealed that the solution contained amino acids, the building blocks of proteins. Of the 20 amino acids used by life, Miller had created several, including glycine, alanine, and aspartic acid. He had also created other organic compounds, a rich mixture of molecules that would never have formed without the energy input from the electric sparks. The Miller-Urey experiment, as it became known, was a sensation. For the first time, scientists had demonstrated that the building blocks of life could form naturally from simple precursors under plausible early Earth conditions. The experiment did not create life, or even anything close to it, but it showed that the first steps on the path from chemistry to biology were achievable through ordinary chemical reactions. Life was not a miracle requiring supernatural intervention. It was a natural outcome of chemistry and physics. In the decades following Miller and Urey's work, thousands of similar experiments have been conducted with variations in the gas mixture, energy source, and conditions. Scientists have used UV light instead of electric sparks, simulating sunlight rather than lightning. They have tried different atmospheric compositions, from highly reducing atmospheres rich in hydrogen to more neutral mixtures dominated by carbon dioxide and nitrogen. They have added minerals to simulate reactions on surfaces. They have used freezing and thawing cycles to simulate ancient ice environments. These experiments have taught us that organic molecule synthesis is robust. While some conditions are more productive than others, amino acids and other organic compounds form under a wide range of scenarios. Even if the early Earth's atmosphere was not as reducing as Miller assumed, organic molecules could still have formed through atmospheric chemistry, reactions in hydrothermal systems, or delivery from space via meteorites and comets. Subsequent experiments have moved beyond synthesizing simple building blocks to assembling them into larger structures. In the 1950s and 60s, Sidney Fox and his colleagues showed that amino acids could be heated to form protoproteins, chains of amino acids linked together. These protoproteins, which Fox called proteinoids, could spontaneously organize into microscopic spheres when added to water. These proteinoid microspheres had some properties reminiscent of cells, with membranes that could selectively allow certain molecules to pass while excluding others. In the 1960s, Juan Oro demonstrated that adenine, one of the nucleotide bases found in DNA and RNA, could be synthesized from hydrogen cyanide and ammonia, both plausible prebiotic molecules. Further work showed that other bases could also form through prebiotic chemistry. Scientists also succeeded in synthesizing sugars, including ribose, the sugar component of RNA, through reactions that could occur on the early Earth. The 1980s brought a revolution with the discovery of catalytic RNA by Thomas Seck and Sidney Altman, working independently. They found that certain RNA molecules, which they called ribozymes, could catalyze chemical reactions much like protein enzymes. Some ribozymes could cut and splice other RNA molecules. Others could even synthesize new RNA using an existing strand as a template. This discovery transformed origin of life research by showing that RNA could serve both as genetic material and as catalyst, potentially solving the chicken and egg problem of which came first. Building on this discovery, researchers have created increasingly sophisticated ribozymes through a process called in vitro evolution. Starting with a large library of random RNA sequences, scientists can select those sequences that have desired properties, such as the ability to bind to a particular molecule or catalyze a particular reaction. These selected sequences are then copied, with some random mutations introduced, and the process repeats. Over many generations, the RNA evolves, with successful variants spreading and unsuccessful ones disappearing. Through in vitro evolution, scientists have created ribozymes that can perform remarkable feats. Some can copy RNA sequences over 100 nucleotides long. Others can synthesize specific molecules or catalyze reactions that nature never evolved. While no ribozyme yet exists that can copy itself completely and accurately, we are getting closer. Each advance demonstrates that self-replication through RNA is not only possible, but achievable through natural processes of variation and selection. The study of lipid vesicles has also progressed dramatically. Researchers have shown that simple fatty acids, which would have been present on the early Earth, spontaneously form vesicles in water. These vesicles can grow by incorporating more lipids from the environment. They can encapsulate other molecules, trapping them inside. And remarkably, they can divide, creating two smaller vesicles from one larger one, when subjected to physical stress such as shear forces from flowing water or osmotic pressure differences. Jack Szostak and his colleagues have created protocells that combine lipid vesicles with self-replicating RNA. In their experiments, RNA molecules enclosed within fatty acid vesicles can undergo copying reactions using building blocks that diffuse through the membrane. As the RNA replicates and accumulates, the vesicle grows to accommodate the additional material. When the vesicle divides, the RNA is distributed to both daughter vesicles, creating a primitive form of reproduction. While far simpler than any modern cell, these protocells demonstrate that the key functions of life, replication, and compartmentalization can be achieved with relatively simple chemistry. Other researchers have focused on recreating primitive metabolism. Günter Wechtershauser proposed that iron-sulfur minerals found at hydrothermal vents could catalyze the reduction of carbon dioxide to organic molecules using chemical energy from the oxidation of iron. Experiments have confirmed that these reactions can occur on mineral surfaces, producing small organic molecules including components of amino acids and sugars. This suggests that a mineral-based metabolism could have preceded and supported the evolution of more complex organic catalysts. More recently, scientists have attempted to integrate replication, metabolism, and compartmentalization into unified systems. These efforts aim to create a minimal artificial cell, the simplest possible system that exhibits all the key properties of life. While complete success remains elusive, significant progress has been made. Researchers can now create vesicles that contain both metabolic enzyme systems and replicating genetic material, allowing the protocell to generate energy, synthesize building blocks, and copy its genome, all within the protective lipid boundary. The goal of creating life in the laboratory is not to prove that life began in exactly the way the experiments suggest. The early Earth conditions were complex and varied, and the actual path to life may have been quite different from any laboratory scenario. Rather, the goal is to demonstrate that life could arise through natural processes, that no miracles or violations of physical law are required. Each successful experiment adds to the catalog of possible pathways from chemistry to biology, expanding our understanding of what is possible. These experiments also reveal the challenges facing the origin of life. Creating individual components is relatively easy. Integrating them into a coherent, evolving system is hard. Getting RNA to replicate efficiently in water is difficult. Synthesizing all the building blocks simultaneously in compatible conditions is challenging. Evolving from simple protocells to cells with the sophistication of even the simplest bacteria seems to require many steps, each individually unlikely. Yet somehow, on the early Earth, it happened. Understanding how remains one of the great challenges of science. Looking forward, the next generation of origin of life experiments will likely use more sophisticated techniques and more realistic conditions. Microfluidic devices can create thousands of tiny compartments, each a potential protocell, allowing researchers to observe many parallel experiments simultaneously. Advanced analytical techniques can detect and characterize molecules present in minute quantities, revealing reaction pathways that would have been invisible to earlier researchers. Computer simulations can model complex chemical networks, predicting which reaction pathways are most likely to lead to self-sustaining systems. Some researchers are pursuing an alternative strategy. Instead of trying to recreate the origin of life, they are trying to create artificial life using different chemistry. If we can design and build a living system from scratch, using molecules and principles of our choosing, it would demonstrate that life is not tied to one specific estry, but is a general property that can arise in multiple ways. This would profoundly influence our thinking about what life is and where we might find it in the universe. Final reflections. The meaning of our origins. We have traveled far together on this journey through deep time, from the formation of the Earth to the emergence of the first cells. We have witnessed the cooling of a molten planet, the formation of the first oceans, the synthesis of organic molecules, and the gradual assembly of these molecules into systems that could grow, replicate, and evolve. We have seen how chemistry became biology, how lifeless matter became living cells, how the universe became aware of itself. This story, remarkable as it is, raises profound questions about existence and meaning. If life arose through natural processes, through ordinary chemistry and physics operating over geological time, what does this say about who we are and why we are here? Some might find this realization troubling, stripping life of its mystery and reducing us to mere chemical reactions. But this perspective, I would argue, is backwards. The natural origin of life does not diminish its wonder but amplifies it. Consider what the origin of life reveals about the universe we inhabit. We live in a cosmos where matter, left to its own devices, organizes itself into ever more complex forms, where atoms forge themselves into molecules, molecules into cells, cells into organisms, organisms into ecosystems and civilizations, where the laws of physics do not merely permit life, but seem almost to encourage it, creating the conditions and providing the mechanisms by which complexity can emerge from simplicity. We are not separate from nature, not souls trapped in mechanical bodies, but integral parts of the natural world. The carbon in your bones was forged in the heart of a dying star. The oxygen you breathe was produced by ancient bacteria learning to harness sunlight. The genetic code in your cells is the same code used by every living thing on Earth, a legacy of our shared ancestor who lived billions of years ago. You are made of the universe, by the universe, in the universe. You are one of the ways the universe experiences itself. The origin of life also reveals the power of time and chance working together with natural law. Given the vastness of time and space, improbable events become probable, even inevitable. A molecule that might form only once in a billion years will form a billion times over a billion years. A combination of circumstances that occurs only once on Earth might occur millions of times across the galaxy. The origin of life required unlikely events, but given the scale of the universe, the unlikely becomes common. This suggests a profound possibility. We may not be alone. The same chemistry operates throughout the cosmos. The same physics governs distant stars and planets. If life arose here through natural processes, it may have arisen elsewhere as well. Perhaps many times, in many places, taking many forms. The universe may be teeming with life, from simple bacteria to complex civilizations, all arising through the same fundamental processes that gave rise to us. Yet even if life is common in the universe, each instance remains precious. You are not diminished by being part of a larger pattern. Every snowflake follows the same physical laws, forms from the same processes, yet each is unique and beautiful. Similarly, life on Earth follows universal laws, but has taken a unique path, shaped by our planet's specific history and environment. The life we know, the life we are part of, is the only example we have, and it deserves our wonder and our protection. The origin of life story also reminds us of our responsibility. We are the descendants of that first cell, inheritors of 4 billion years of evolution, bearers of the flame that first sparked in an ancient ocean. That flame has burned continuously from the first protocell to you, never extinguished through ice ages and impact events, mass extinctions, and dramatic changes. We have a responsibility to ensure it continues burning, that the chain of life that began so long ago does not end with us. This means protecting the biosphere that sustains us, the only home for life we know. It means preserving the diversity of species that share this planet with us, each one a unique experiment in biology, each one irreplaceable once lost. It means thinking carefully about our impact on the environment and the legacy we leave for future generations. We are not just observers of life's story, but active participants, and our choices matter. As you prepare for sleep, let these thoughts settle gently in your mind. You are a temporary arrangement of matter and energy, a pattern that persists for a time and then dissipates back into the universe. But while you persist, you are a wonder. You are consciousness emerging from chemistry, thought arising from neurons, awareness blooming from atoms. You are the universe contemplating itself, asking questions about its own origins, marveling at its own existence. In the grand sweep of cosmic history, your individual life is brief, a flash of lightning in the darkness. But it is your flash, your moment to experience existence, to feel and think and wonder. You are connected to everything that has ever lived and everything that will ever live, part of an unbroken chain stretching back 4 billion years and forward into an unknowable future. That connection, that continuity, is something profound and beautiful, something worth celebrating. So rest now, knowing that you are part of the greatest story ever told. The story of how a lifeless planet became a living world. The story of how chemistry became biology, how matter became mind, how the universe became aware. The story of life itself, in all its wonder and complexity, its beauty and mystery. You are not just reading this story, you are living it, embodying it, continuing it. And in that, there is meaning enough for any life, however brief. Dream well, child of the cosmos. Dream of ancient oceans and the first dawn of life. Dream of molecules dancing in the darkness, finding each other, joining together. Dream of protocells dividing in warm tidal pools, of RNA replicating in mineral pores, of the first metabolisms flickering to life around volcanic vents. Dream of 4 billion years of evolution, of all the ancestors who survived so that you could be here now. Dream of the future, of where life might go, of what wonders might yet emerge. And rest in the knowledge that you are exactly where you should be, when you should be, part of an unfolding story larger than any individual life yet made meaningful by each individual life. Good night. Postscript. As you drift into sleep, carrying these ideas with you into the realm of dreams, remember that the origin of life is not just ancient history. It is ongoing. Every time a child is born, molecules that once were separate come together to form a new living being, a new instance of the pattern that first emerged 4 billion years ago. Every time a seed germinates, chemistry becomes biology once again, as water activates the molecular machinery stored in dormant form. The miracle of life's origin is not something that happened once in the distant past. It is happening now, all around us, in every new life that begins. And perhaps most remarkably, life continues to evolve. Just as the first cells evolved from simpler protocells, just as multicellular organisms evolved from single cells, life continues to explore new possibilities, to adapt to new environments, to discover new ways of being alive. We stand not at the end of evolution, but in the middle, with millions or billions of years of future evolution ahead. What new forms of life might emerge? What new complexities might evolve? What might life become, given enough time? These are questions we can only imagine, but they remind us that the story of life is still being written, and we are privileged to be part of it, if only for our brief moment in the vast sweep of time. Sleep peacefully in this knowledge.